and we talked about the cranial nerves. How many pairs of cranial nerves are there? 12 pairs of cranial nerves, and you should know them in order and what their number is, what their major function is, and the packet that I assigned to you, I think, would help you a great deal with that. So we're going to kind of go through that section really quick, but we'll go more slowly on areas that I think will be more important for you guys. So this is the brain. The brain weighs about 3 pounds. Of course, I like to think that my brain weighs about 10. But, typical structure of the brain, there's 4 major parts. What's this large part here? Cerebrum. Okay, in the back of the cerebrum here, cerebellum. Good. This area, brainstem. And if I was to split this without dropping it totally, do that. And this center region in here, not the diencephalon. Okay. In the diencephalon, we have the thalamus and hypothalamus. Here is the brain stem, kind of white colored area in the model. At the very top is called the midbrain. And the pons and the medulla oblongata are also part of that brain stem. Our major focus is really going to be on the cerebrum. And so we'll talk mostly about, this is the mid-sagittal section through the brain. You can see the cerebrum nicely here. The corpus callosum, this area of white matter, of commissural fibers, running across from both cerebral hemispheres, so the right side of your brain is communicating with the left side of your brain. Of course, here's the cerebellum, and then the brain stem, the pons. The brain stem, the medulla oblongata, is connected with the spinal cord. They are as one continuous group of nervous tissue. So the axons that we were following last time, moving up and down the spinal cord, those axons are moving up the spinal cord into particular areas within the brain. The motor information's coming down from the brain. The axons, again, moving through the spinal cord, from the brain down through the spinal cord to a particular muscle within the body for those actions to take place. We've looked a little bit at the pituitary gland here hanging down from the hypothalamus, mainly because the pituitary gland is one of those glands that's going to release hormones with the endocrine system. And we talked within the skin about the difference between the exocrine glands and the endocrine glands. Exocrine glands being those like sweat glands and sebaceous glands and goblet cells. But here we would look more at the endocrine gland, the pituitary gland, and its ability to secrete hormones within the bloodstream. So we'll talk a little about the blood supply to the brain and the unique blood supply around the pituitary gland itself. So that when the pituitary gland is releasing hormones into the blood, those hormones are moving directly into the bloodstream and will circulate quickly throughout the body. We've looked at the meninges around the spinal cord. There's 3 meninges around the spinal cord. The pia mater is the deepest of all 3 of the meninges. The arachnoid mater, and what's the most superficial? The dura mater. We have the same meninges here around the brain. They are in the same orientation. So pia mater is the deepest, and then arachnoid, and then dura mater is the thickest. When we do the dissection later today, you will be able to see the dura mater, and in fact, I'll show you that you can peel off the dura mater. It's almost like thick saran wrap wrapped around the brain. And you will need to peel off that meninges in order to actually dissect or cut the brain into pieces.
So the meninges in the brain, it's the same as the spinal cord?
It's the same as the spinal cord, yes. Yep. So if we were looking here at the outer surface of the brain, this section of the brain is more of a coronal plane or a frontal plane that you're looking at. You can see the are all around the outside here of the brain. You can also see that they are moving within to kind of separate the cerebrum from the cerebellum. It's easier to show you with this model because I can actually move it apart a little bit. You see how this tissue is not joined here? So I have the cerebrum and the cerebellum, and I have a space in between. If we were able to see the meninges around this area, the meninges would actually move in between this space. It's as if I take this card and put it right in here. That's the way the meninges would go. Okay. This is called the transverse fissure within the brain. And the area of the meninges moving in this transverse fissure is called the tentorium cerebelli. Highlight it on the screen so you can see that right here. Tentorium cerebelli. It's just the meninges moving within this transverse fissure here in between the cerebrum and the cerebellum, the back of the brain. The other thing that's noticeable from this diagram is the ventricles within the brain. These are equal to the central canal in the spinal cord. What was flowing within the central canal? Good. Cerebrospinal fluid. So here's the central canal within the spinal cord coming up this way, and that will continue on moving upwards into the ventricles. The ventricles themselves are basically spaces, cavities, where cerebrospinal fluid is flowing through. The cerebrospinal fluid is produced from cells here in the choroid plexus. This is kind of red colored area right here. The choroid plexus is composed of neuroglia cells. Since we're in the central nervous system, do you remember the type of neuroglia cell that produced cerebrospinal fluid? Epididymal cells. Good. The epididymal cells, they look like, almost like columnar epithelial cells. Those are the cells that were lining these blood vessels that are producing this cerebrospinal fluid. So these darker red areas in the diagram represent those epididymal cells, and the group of them is the choroid plexus. On some of the brain models, if you look at the brain from the inside in the mid-sagittal section, you can see a blue area or a darker area. It's right here below the corpus callosum. That is the choroid plexus. Okay, so this area here that is purple is the choroid plexus. It is the same as this one here. The only difference is here we're looking at a mid-sagittal plane. Here we're looking at a frontal or a coronal plane. But we're still looking at that choroid plexus, the epididymal cells there, producing the cerebrospinal fluid. Melissa.
So all the choroid plexus is a group of them?
Yep. So if these epididymal cells are producing cerebrospinal fluid, where's the fluid going to flow? Where is it going to empty into? It's going to empty into the ventricles, these spaces here. And these are the lateral ventricles. There's one in each cerebral hemisphere. In back of this area is the lateral ventricle for this half of the brain that I'm holding. Some of the brains actually have that area opened up. If you have one of those, certainly hold it up for everyone to see. This one is like that. Okay, so you can see this open space right here within this model. That open space is the lateral ventricle. So that's where the epididymal cells will be releasing the cerebrospinal fluid and will be filled with that cerebrospinal fluid. So if we're in the lateral ventricles here, producing cerebrospinal fluid, the fluid is going to go down this way into an area called the third ventricle. It's going to pass through this small channel, which is the cerebral aqueduct, and on into the fourth ventricle, which is very close to the cerebellum. Just show it to you on here. So we're inside the lateral ventricle, cerebrospinal fluid. We're moving from the lateral ventricle down this way into the third ventricle and the cerebral aqueduct, and here into the fourth ventricle. Can you all see that space? Okay, so this is the fourth ventricle. It's very close to the cerebellum. It's equal to the space that they're showing you right here. From this area of the fourth ventricle, the cerebrospinal fluid is going to flow down through the central canal of the spinal cord. Now remember, cerebrospinal fluid also moves within the subarachnoid space, and the subarachnoid space is shown here in that kind of darker tan color, all the way around the spinal cord and around the brain. As I said before, the same meninges that we've looked at around the spinal cord are around the brain in the same order. So if the cerebrospinal fluid is moving here in the subarachnoid space, that's going to be continuous with the subarachnoid space all the way around the brain. So we have this flowing of cerebrospinal fluid lubricating both the brain and the spinal cord, moving on the outside areas of both the brain and the spinal cord and the subarachnoid space, but also moving on the inside of the spinal cord and the central canal, and on the inside of the brain within the ventricles of the brain. So, I'll get on to the picture because it's a little bit easier to talk about it. So here's another view of the ventricles within the brain. And you can see from this view, the ventricles filled with this blue colored fluid. In reality, cerebrospinal fluid is really not blue. It's pretty much clear. Okay? If it is anything other than clear, then that's an indicator of a problem, of an infection or a problem. With this C-shaped area here, this is the lateral ventricle. You can more easily see the connection between the lateral ventricle and the third ventricle. You picture all of this light blue as fluid-filled areas. The lateral ventricles connect to the third ventricle through a little foramen, a little tube, which is the interventricular foramen. Interventricular, in between the ventricles. Foramen is a hole. So we're bringing the cerebrospinal fluid from the lateral ventricles through the interventricular foramen into the third ventricle that you can see here, and then down through the cerebral aqueduct, another little channel, into the fourth ventricle. The fourth ventricle, again, close to the cerebellum, is going to be continuous with the central canal within the spinal cord. And you'll see this blue color move all the way down throughout the spinal cord. At the same time that the fluid is moving down through the spinal cord, it is moving through foramen or holes, channels on either side of the fourth ventricle, which are called lateral apertures, and that's going to bring the fluid back up and around the brain. So it's not only just going straight down, it's also kind of going down and up and around the brain. That's what they're trying to show you right here with the lateral apertures. So here's another view of that, an anterior view. You can see both halves of the cerebrum together facing you, and both of the lateral ventricles facing you, the point at which they're draining in to the third ventricle. You can see very nicely the interventricular foramen there. To me, this almost looks like bull horns. So if that helps you to remember it, the lateral ventricles are like the horns of the bull. They're on each side. Okay, so you have one in each cerebral hemisphere. There is only one third ventricle. There's really only one fourth ventricle. One cerebral aqueduct, and one central canal, but two lateral ventricles. So what's the purpose of this cerebrospinal fluid? Lubricate the spinal cord, lubricate the brain. What else? Protection. Good. So it's kind of like packing something very delicate in bubble wrap, right? Where the bubble wrap is plastic, but what's inside the plastic? There's little pockets of air. So you almost like have this bubble wrap around the spinal cord and around the brain. But as well, there's nutrients within this bubble wrapping, right? There's nutrients within the cerebrospinal fluid. There's electrolytes and so on, water within that fluid to lubricate the tissue. We don't want the tissue to dry out. Okay, all of our cells function best in a watery environment. If they are dried, if they are dehydrated, then the nervous system is really not going to function. This goes through the flow of cerebrospinal fluid. We've done it a few times. But the right-hand diagram is very nice because you can clearly see the choroid plexus here, these red areas, ependymal cells around the blood vessels, and you can see nicely the tentorium cerebelli, which is this invagination of the meninges in the transverse fissure right here. So you can see the blue moving all the way around the outer surface of the cerebellum and the cerebrum. All of this blue is that cerebrospinal fluid. So here's our cerebrospinal fluid. You can see the areas where it is traveling by looking at the black arrows, and opposed of all of these other areas here where the white arrows are showing you the blood flow throughout the tissue. I think it was Paula that asked me earlier, over here you have these pulse points within the outside of your brain. You obviously have blood flow around your brain. If you didn't have blood flow to your brain, you wouldn't be thinking, you wouldn't be feeling, sitting here, responding. And that the brain feeds off of the sugar within your bloodstream. So the sugars, the carbohydrates, are a source of energy for your body, but also a source of energy for your nerves to function. So if you don't eat properly, your brain is not going to function properly. If you don't sleep a certain number of hours a day, your brain's not going to function properly. It's actually when you sleep that your brain has more of a chance to kind of put things where they're supposed to be, set your memories in order, or set everything that you're learning in its proper place within your, what we call long-term memory, so you're going to hold on to it. So if you try to cram for a test, we've all been there, you might remember a few things the next day, but the day after that, you're not going to remember any of that. So try not to cram, try not to do the last minute studying, try to get sleep because that will allow you to store things long term. Try to drink, try to make sure that you eat. I know, this might be like the little mom lecture to you, but if you want your brain to function properly and hold on to this information, this is what you should do. So we've talked, I think, quite a bit now about moving the cerebrospinal fluid and lubricating the brain and feeding the brain with the nutrients from cerebrospinal fluid, but also from the blood. You can see a nice close-up here of the different areas of the meninges wrapping around the brain. The green-colored area is the outermost meninge, which is the dura mater. That's this green area here. This pink-colored line is the arachnoid mater. And then the red, the very thin red line, represents the pia mater. And in between the pia mater and the arachnoid mater is that subarachnoid space. See that right here? You can see where the the name arachnoid comes from, because it almost looks like it has spider webs throughout this space in this area. So this is where the cerebrospinal fluid is flowing within this subarachnoid space. Blood, on the other hand, is here. And in the areas of the arachnoid mater, we have these enlarged, and they almost look like broccoli florets. These are the arachnoid villi. And it's the arachnoid villi that are going to be responsible for reabsorbing the cerebrospinal fluid. The rate at which the cerebrospinal fluid is produced is pretty much equal to the rate at which it is reabsorbed by the arachnoid villi. And I'll flip back again to the previous picture so you can see that a little more clearly. Again, we're producing the cerebrospinal fluid within the choroid plexus. See the very nice choroid plexus here within the lateral ventricles. It's circulating around the brain and around the spinal cord, and we are reabsorbing that back into our bloodstream with those arachnoid villi, with the structures that I said look like broccoli. And you will see those throughout this entire area here. And though they're not drawing them in many areas, you see that one has been drawn here and here and here, but they are throughout this entire area of the top of the brain, the top of the cerebrum. So we're recirculating that fluid, we're reabsorbing it back into our bloodstream, and the choroid plexus, epidermal cells are producing more, and then we're reabsorbing it back in, and we're producing more, and that keeps going. So it's not as if you are kind of sitting within the bath water for days on end. The bath water is constantly being changed. Does anyone remember the type of neuroglia cell that was responsible for finding pathogens and eliminating pathogens within the brain tissue? Yes, those are the microglia. Okay, so aside from us constantly reabsorbing the fluid and recirculating new fluid, we have these microglia that are detecting any pathogens that might be there and hopefully responding and destroying them. Flip back one more time. Okay, so this same area here, this tentorium cerebelli, you can't see right here, but it is there. They're only showing you the ventricles on the inside. You cannot see here, again, they're only showing you the ventricles on the inside, but you can see right here. It's going to be one of those hard structures to point to, especially when we do the dissection of the brain later on today. So, with that structure, that might be one of the times that I will use a diagram, and I would use one of the ones that I just showed you to ask you that structure. So again, right here is the tentorium cerebelli.
Straight sinus?
What's that?
It says straight sinus.
The sinus is the actual blood flow within that area. So if you were looking at the blood vessel in the area, that's the straight sinus. But the meninges moving in the transverse fissure, that's the tentorium cerebelli. At any point where you see the light blue here, that's fluid. And essentially that's fluid that we're recirculating throughout the blood vessels, throughout your blood. At any point where you see the darker tan color, that's also fluid, and that's representing the cerebrospinal fluid. Sinus is just an enlarged blood vessel. All Alright, so we will look then at the major parts of the brain and talk a little bit about the functions of each of the parts. So if we start with the medulla oblongata, again, that's a continuation of the spinal cord. The medulla oblongata consists of the nuclei of 5 of the 12 pairs of cranial nerves. The cranial nerves are extending from this area here. One of the most important functions of the medulla oblongata is the cardiovascular function. It helps to regulate the force of your heart rate. So if your heart is beating very slow and you're very calm, or you see something, your heart starts to beat much faster, it's kind of racing, that's a response from the medulla oblongata, right? We're trying to regulate the heart rate in response to the stimuli around us. The other major function of the medulla oblongata is regulating the diameter of blood vessels. Not necessarily here within the brain, but in the cardiovascular area. So within the aorta, around the heart, major cardiac vessels, the carotid arteries within the neck. We're trying to regulate the diameter of those blood vessels feeding the heart itself,
Based on.
not the brain, based on external stimuli.
What it's called, the blood. Whether it's water.
Yeah, I would say more on whether there is enough oxygen or too much carbon dioxide. And then in response to that, obviously the blood vessels are going to change in diameter, but your breathing rates are going to change. If you have too much carbon dioxide, you're going to start to breathe more rapidly so that you exhale that carbon dioxide more quickly. Or if you don't have enough oxygen, then you're going to start to inhale more deeply. You know, your mom says take deep breaths to bring that oxygen level up. But more regulated is the amount of CO2 than the amount of oxygen.
You said it was an extension of the spinal cord.
The medulla oblongata, yeah, the two of them are connected. So the medulla oblongata is the last region here, and from that you can see the spinal cord.
So that's where they connect.
They're connecting with each other. So what we would think of as involuntary activities, the regulation of our heart, the beating of our heart, how fast or how slow, the rate of our breathing, how quick we're breathing or how slow we're breathing, if we're breathing at all, right? All of that is the job of the medulla oblongata to regulate. Okay, this is going to be interesting when we talk about this special sensory pathways. We will look more closely at the medulla oblongata, and you can see within the medulla that there is an area of which the axons cross over from one side of the body to the other side. This is called decussation of pyramids. You can see it here in yellow. Okay, this is the area of the spinal cord. If you want to think about the axons coming up the elevator from the spinal cord, if we're coming up this area here, the axons are going to cross to the opposite side of the brain. Same thing on this side. The axons coming up the spinal cord here on this side are going to cross in the medulla oblongata and move to the opposite side of the brain. When we say axons cross, we say they decussate. That's decussation. It's just the crossing over of the axons going to opposite sides. With particular pathways, this happens. Many of the pathways, in fact, will cross from one side of the body to the opposite side, and the crossing of them happens within the medulla oblongata. It happens within the pyramids. If you look at the medulla oblongata from the outside, if you were to look at the model of the brain, you really wouldn't be able to see that just from the model. But we can see it from the outside. mentioned the medulla oblongata is the site where 5 cranial nerves, nuclei, arise. And you can see the nuclei of the 5 cranial nerves here. We're looking at cranial nerve 8, 9, 10, 11, and 12. Cranial nerve 8, which is right here, vestibulocochlear. 9, glossopharyngeal. 10 is vagus. 11, accessory. And 12, which is hypoglossal, they're not showing you because it would basically hide all of this. Okay, but 8 through 12 are originate from the area of the medulla oblongata, basically from this point down here. That's all medulla oblongata.
What about 7?
7 does not originate in the medulla oblongata. It's actually a little bit higher than the medulla. 7 is the facial nerve.
That's not part of the.
It's not included in the medulla, no. We talked a little bit about the 12 pairs of cranial nerves last time, and I wrote the mnemonic device on the board. So I'm hoping that you remember, right, that this is 8, 9, 10, 11, yes. You're with me, yeah. It starts from 8. Well, for the medulla, yes. 8 through 12 is in the medulla. Some of you have brain models of which the cranial nerves are numbered. So, you know, if you see cranial nerve 8, you'll notice it is very close to 7, but it is below the area of the pons right here. So it's kind of from here to here, where you will be able to see cranial nerves 8 through 12. On your lab practical and on your quiz, you will have to tell me where is cranial nerve 1, where is cranial nerve 2, and so on. So if I am somewhere within the area of the medulla oblongata, at least you can narrow it down to anywhere between 8 and 12. They also do go in order. Did you notice this? Mm-hmm. From 1, 2, 3, they're pretty much in order downwards. 12 is the correct one. Okay. So since the medulla oblongata is responsible for regulating heart rate and breathing rates, if you were to get hit on the back of the head in the area of the medulla oblongata, you would most likely lose consciousness for a certain amount of time. Okay, it's, you know, big sensation to the medulla oblongata. It's going to shock the neurons within the area, and you'll essentially pass out. You've seen in those crime shows, someone hits you on the back of the head, and they, you know, they fall down. Yeah, that's because of the medulla. Okay, the pons is the next area, kind of moving upwards in the brain stem. So here's the pons. It looks like an enlarged oval. Kind of on top of the medulla oblongata, right here. This is the pons. The pons also is a control center for the regulation of breathing. Our respiratory centers within the pons. In the pons is the nuclei of cranial nerves 5 through 7. So this is where the facial nerve comes in. Odette asked me about a few minutes ago. So cranial nerve 5. What's cranial nerve 5? Good. Trigeminal. 6. Abducens. And 7, I already just said, facial nerves. Those are all from the pons. The diagram here is the pons right here, helping us to control breathing, the pneumotaxic and apnoistic areas. We'll come back to that with the respiratory system areas where we are regulating the breathing. Okay, midbrain. I have a highlight. Yeah, I do. So the midbrain is this section here. It's in the midbrain where you can see the cerebral aqueduct. Okay, here's the lateral ventricles, and we're moving down here cerebral aqueduct on into the fourth ventricle, across the cerebellum. Not really much to say about that, other than the cross section that you can see here, which is the transverse section through the midbrain. You can see the red nucleus and the substantia nigra, which is helping to control subconscious muscle activity. This area here that is highlighted in red, and this darker area here that's colored in black. The colorations here are actually pretty true to the brain tissue when you do the dissection. You might see in this area the center tissue in the midbrain is a kind of reddish color, and that's because of the amount of hemoglobin there and the amount of iron that's within the hemoglobin that creates this kind of red coloration. The black coloration as well. This is a kind of darker mass of tissue here. This black area this is why we call it the substantia nigra for a black color. This is what's basically allowing you to sit still, right, and a normal kind of action. Individuals that are sitting and then constantly like fidgeting, you ever know, so that they're constantly shaking their leg as they're sitting there, okay? You know, we're kind of suppressing, normally we are, suppressing that activity by these areas of the brain. Okay, so someone that is kind of fidgeting like this is not as, I don't want to say it like that, but it's not suppressing the activities as much as maybe somebody else might be doing. The peduncles. Peduncles are the kind of two enlarged areas. When we cut through the midbrain and the transverse section, you would be able to see that. We can try to do a section like this through the brain in the dissection. I don't believe I have any models of the brain cut in this way to show you those. But so if we were to take the brain like this, if I had both pieces that moved, it would be helpful, but I don't. And we were to hold the cerebrum and pull down on the cerebellum, that's what they're doing right here. Okay. They're holding the cerebrum still, pulling down on the cerebellum to the point where you could see the bumps, the indentations. See the bumps between the two structures. So in the midbrain area, right here, you can see what we call the tectum. It's composed of 4 bumps. Corpora quadrigemina is the entire 4 of them together. There's 2 superior bumps, those are the superior colliculi, and the 2 inferior bumps are the inferior colliculi. These 4 bumps in between the cerebellum and the cerebrum, kind of pulling them apart. You can see this from the back of the brain. It's called the corpora quadrigemina. And it's these 4 bumps right here. Here's the 2 superior colliculi and the 2 inferior colliculi. They're responsible for allowing you to turn towards something that you hear or turn towards something that you see. So if you see, I see someone coming in the door at the corner of my eye, I'm going to turn towards that. That's because of the corpora quadrigemina. Or I hear something on the other side of the room, I'm going to turn towards that. It's because of the corpora quadrigemina. If a person can't hear, then part of it, it's still functioning, it's just the sensation of them hearing isn't getting to the brain to be processed. But they, assuming that they still have sight, they can still turn towards the sight. Many individuals that are deaf, that can't hear, where they wouldn't hear the phone ring. The phone has a light on it, and so they would be able to see the light. We will see this on the dissection. You will be able to see the superior colliculi and the inferior colliculi. On some of the models, not on mine, unfortunately, it's not very nice, but on some of the models, if you look at the very top of the cerebellum, you can kind of see these bumps. You can see it maybe better from this view. See that? There's 2 bumps here. These 2 bumps, that's the corpora quadrigemina. So this top bump is the superior colliculus, and the bottom is the inferior. The other way that's going to be helpful for you to recognize these structures is you will be able to see the pineal gland within the brain. A few of you saw this last time. The pineal gland is right back here. Okay, in the brain tissue, it is a darker colored tissue. It looks like a little green pea in the back of the brain. So this is a darker area here. You look below the pineal gland, then you will be able to see the corpora quadrigemina. On this model, it's this white area that I'm pointing to here. And there should be 2 bumps, a top bump and a bottom bump. This model really isn't such a great one to ask that question, but you will be able to see it nicely on the dissections. Yes, it's just another name for corpora quadrigemina. Yeah, I know, it's easier to spell. Yes. Yeah. But you should know superior colliculi and inferior colliculi. Because I will put the pointer on one or the other, essentially. And then in the diagram, you can see the pineal gland right here. That's going to be kind of a hard thing to see from this angle. But again, if you look for the darker colored tissue, I think that would help you. So we have reticular activating system, reticular formation. Reticular activating system is the area of the brain of which wakes you up, right? You know when you're sleeping, you don't hear anything. You're not seeing anything, obviously. You're not responding to visual sensations or auditory sensations. Your brain is actually still processing sounds all around you. So your brain still hears the dishwasher going and the cat jumping off the bed and whatever. It's just saying, oh, that's not important right now. Just ignore it. That's what your brain is saying. But if you were to, set your alarm clock goes off, or the television for some reason just goes on, husband turns the TV on at night or whatever. These things are kind of set to wake you up, and they activate the reticular activating system. So the fire alarm goes off, and you immediately wake up. It's at the point where the brain says, oh no, she needs to be aware of this, so I'm going to wake her up. All of these alert situations activate the reticular activating system.
And is that send action potential then?
You're really still processing action potentials. It's just your brain is kind of ignoring the ones that it doesn't think are important. And at the point where your alarm goes off, the brain says, oh, she has to get up now, then it's going to send another action potential series of action potentials to get alert to it.
Your brain just ignores. Like screw that.
Yeah, pretty much. So, you talk in your sleep, which can be the case for some people. You probably make sense. It's just you don't realize that you're talking because your brain's ignoring it. Versus the other person will realize that, I'm sure. But okay, keeping you awake. So aside from waking you up, aside from the alarm clock waking you up, it's the reticular activating system that's keeping you awake. constantly detecting stimuli and responding to them. You see the lights are on. You're moving around, things are moving around you. You're processing all of this information, and so you want to stay awake. Your body wants to keep you awake with this. Medulla pons midbrain, that's the location of the reticular activating system. All this kind of center area of the brain stem, keeping you awake, keeping you conscious. All right, cerebellum. Looking at the cerebellum. Cerebellum is the base of the brain. Unfortunately, I don't have a piece of one that's detached. Use this one though. The cerebellum is unique because the gray matter and the white matter within the cerebellum. The white matter is obviously deep. Gray matter is more superficial, just like the other areas of the brain. However, within the cerebellum, the white matter has this kind of tree-like arrangement to it. It looks like the trunk of a tree and the many branches of the tree. And you can see that when the cerebellum has been cut in half. This white area of tissue in the cerebellum is called the arbor vitae. Arbor for tree, vitae for life, so it's the tree of life. Tree of life. Arbor vitae is the white area. The folia, I think of foliage from outside, the kind of greenery, the leaves of the tree, that's the gray matter within the cerebellum. So the tree itself would be the white area, the arbor vitae, but around that, all of the leaves, all of the gray matter, that's the folia. I think foliage, but its proper term is folia. And here's another view of the cerebellum. It is intact. They haven't cut it in half. In other words, they haven't done a mid-sagittal section through the cerebellum. So you see the entire cerebellum, both hemispheres, like this. In between the two cerebellar hemispheres is an area that looks like a worm, and that's called the vermis. The vermis.
The vermis divides it, or just the area?
It's just the area in the middle. I don't want to say divides because it's really one piece. Kind of like this worm in the middle of the two. So we have two cerebellar hemispheres, just like we have two cerebral hemispheres. Two kind of halves of the cerebellum. And if we were to do a mid-sagittal section, you'd see the arbor vitae and folia in both of those halves. So again, cerebellums for muscle contractions, allowing you to stand up, allowing you to maintain balance and equilibrium. It's all the cerebellum. Not only if they hit you on the back of the head, might you stop breathing and pass out because they're hitting your medulla oblongata. They're probably hitting the cerebellum at the same time, and you're going to kind of fall over. Lose your sense of balance. I've already showed you the transverse fissure, but I will show it to you again. Okay, the cerebrum here, the transverse fissure is right here between the cerebrum and the cerebellum. So here's the transverse fissure. That's where I was mentioning the tentorium cerebelli before, which is the meninges kind of going within that transverse fissure. You can see it here just because they're kind of pulling the two apart. This gap, this space here, is the transverse fissure.
Isn't there another name for that?
The tentorium cerebelli is the meninges within there.
So the fissure is just a space?
The fissure is just a space, it's a groove, yeah. On this diagram, you can clearly see the arbor vitae, the tree of life, the white matter in the cerebellum, and then the gray matter, which is here, more superficial, you can see the folia. bumps here, this is the corpora quadrigemina that we were looking at within the midbrain, the superior colliculus and the inferior. So this section from here up, this square here, that's all the midbrain. The cerebellum is just this point right here, just this part. So if we were looking at just the cerebellum itself, you would be able to see the different regions in the cerebellum, these enlarged areas. You can see one right here and one over here. These are the peduncles. Just like you saw before. Peduncles. I know, it's a weird word. It's a weird word, right? So they're just kind of protrusions within the area of the cerebellum in this case. So there's one right here. There's another right here. You have to look at the cerebellum. With each of these cerebellar peduncles, you'll see information being carried to and from the brain stem. So if we want to communicate between the brain stem and the cerebellum, those fibers, the axons, would be moving within the peduncles. Road for the axon. Sensory fibers will move in to that area, and motor fibers as well, meaning your balance and stature will be traveling within those peduncles. So within these kind of enlarged areas here, there's sensory neurons, sensory axons moving, and there's motor axons. Motor information moving. On this one? Yep. I had on this one. This one right here. It's kind of this enlarged area here. You got it. Not the vermis, which is in the middle, but it's right next to that. Good. Okay, diencephalon. The diencephalon, again, is the center area. What kind of section is this? Good, it's a frontal section, frontal plane. Okay. You can see the lateral ventricles we were looking at before. These two areas here. So all of this in the center, this is the diencephalon. The kind of largest part of the diencephalon is the thalamus. Below the thalamus is the hypothalamus. Below that is the pineal gland. Pituitary gland. Sorry. So the thalamus, the hypothalamus, the pituitary gland, the epithalamus, these are all a part of the diencephalon. The thalamus is this area here that's highlighted in orange. To me, it looks like two little pieces of mandarin oranges. Okay. These colors are all just, done by the artist. Your brain's not colored this way. So here's the thalamus right here. These two kind of slices of mandarin orange. And then below that, in the teal ovals or circles, this area here, that's the hypothalamus. On the brain sections that we have, we don't have one that's like this one. On your brain sections, this area here is the thalamus. And then below that, all these words right here, that's the hypothalamus. The other easy way for you to find it is if you have the pituitary gland hanging from your model, you look above the pituitary gland, that's the hypothalamus, and then above that, that's the thalamus. So we're below the corpus callosum now. So between the corpus callosum and the pituitary gland, you'll find these. Thalamus is the first kind of larger area. Often it looks like an oval with a little dot in the middle of it.
Just that oval with the dot.
That's the thalamus. And then right below that is the hypothalamus. Below that is the pituitary gland, which I don't have on my brain. My brain's gonna have problems. That's the hypothalamus. And when give you a letter or a number, but they're all lettered and numbered differently. Some of them are bought from different companies. So. The diencephalon is this whole area that I've just colored in red. So the thalamus we'll talk more about as well when we get to the sensory pathways, sensory motor and integrative pathways, because there are many pathways that will synapse within the thalamus. I think I've mentioned this before. If the pathway synapses within the thalamus, we're consciously aware of the actions of that pathway, of the sensations coming in and the information effect going outwards.
It affects consciousness? No?
If we're consciously aware of the sensation going on, then we know it's synapsing within the thalamus. If we're not consciously aware of it, it's not synapsing within the thalamus.
So it could be like prick your finger or something.
It will synapse your thalamus.
So if you don't feel it.
If you're not consciously aware of it, if your heart rate has changed because maybe you're dreaming or something like that.
Okay.
It's like an unconscious or subconscious thing. Thalamic nuclei, these are all the different areas within the thalamus and all the different areas within the hypothalamus. Your book has a picture of all these different colored areas. You don't need to know it. It's too much information. Each of these separate areas of the thalamus is responsible for particular activities. Some for remembering, some for sending information to the basal ganglia and coordinating with the basal ganglia, some for sensing tastes and visual stimuli and things like you don't need to know all of that. The epithalamus, what you should know about the epithalamus, okay, epi, what's epi? Above. So, we were looking at the thalamus. Above the area of the thalamus is where the pineal gland is located. And that's the structure that you should know for the epithalamus, the pineal gland. The pineal gland is right back here.
So we should
Pineal.
The pineal gland is in the epithalamus.
You should, yes. The pineal gland is in the area of the epithalamus. It's also a part of the diencephalon because it's within the epithalamus. The pineal gland, pine, like a pine tree, pineal, is the one that I said was going to be dark and look like a little pea, like a green pea within the brain tissue. It'll be easy to spot in the dissection. The pineal gland produces the hormone melatonin, which some of you got confused with melanin, the pigment within the skin. Melatonin is the hormone. Melanin is the pigment in the skin from the melanocytes. The melatonin is the hormone that is responsible for regulating your sleep and wake cycles. When you're going to get sleepy, when you're going to wake up, and it's all regulated with the amount of light. In darker situations, you get more sleepy. In light situations and bright light situations, you're more awake because the amount of melatonin produced from the pineal gland is different in each situation. So now that it is starting to get darker earlier, you should be getting more sleepy earlier. Like you want to go to bed at 8 o'clock. When normally you didn't get tired until 10. Right? And that's because of the pineal gland. And my circle's a little bit off there, sorry. Okay, circumventricular organs. Circumventricular organs are CEO. Major one being the blood-brain barrier. Major point here. Blood-brain barrier we had talked about with the neuroglia cells. Remember we're looking in chapter 12 with nervous tissue. If you remember the cells which were the astrocytes, the neuroglia cells, that had these extensions on them that would wrap around the blood vessels. And they would form the BBB, the blood-brain barrier, to prevent any pathogens that were in your blood from perhaps going into your brain tissue and affecting your brain. There are regions within the brain that do not have the BBB. They don't have those astrocytes moving around the blood vessels, and that's what we call the circumventricular organs. It's areas that are kind of vitally important to us, but also need to be able to detect what's within the blood and release substances into the blood. There can't be a barrier between those regions of the brain and the blood. There needs to be an ease of access between the areas of the brain and the blood. So the circumventricular organs are areas that do not have BBB. They don't have BBB. Since they do not have BBB, that means that if we have an infection within the blood, the infection could move from the blood into those areas of the brain. Still with me? Yes, kind of? So areas that, the pituitary gland that have to release hormones into the bloodstream, it wouldn't make sense for us to have BBB around the blood vessels in the pituitary gland, because then how are we going to put the hormone in the blood, right? Kind of with me? Right? If we have the pituitary gland and we want to release hormones into the bloodstream, but yet around the blood vessel we had the wall of astrocytes, how are we going to get the hormones into the bloodstream? It wouldn't be possible. So we can't have that protection in these certain areas where the CVO, where the circumventricular organs are. The pineal gland is one of those areas. Melatonin is being released. And the pituitary gland is another one. Hypothalamus as well, but I think it's easier to understand the functioning of it with the pituitary gland. If we had BBB around blood vessels, around the pituitary gland, then there would be no way for us to get follicle stimulating hormone to the ovary and estrogen to the ovary and growth hormone throughout the body, things like that.
What do you mean at the bottom without anxious?
I was going to just talk about that, yeah. So the BBB in itself, like I said, is a defense mechanism for us. It's one of our ways that will prevent a virus or an infection, pathogen, bacteria, something like that, from getting into the brain tissue. But let's say that substance is traveling within your body, it's traveling within your bloodstream. It might not be able to get into this particular area of the cerebrum because the BBB is there, but when it comes across throughout the blood system to the pituitary gland or to the hypothalamus, it then had access. It then had like an open door. So that the HIV virus is one of those that will definitely take advantage of that. And in advanced stages of HIV, the virus will not only spread throughout your immune system and attack your white blood cells, which are CD4 cells, but the white blood cells are part of your blood circulating throughout your body, will then have access to the tissue within your brain, within the hypothalamus, and it will set up shop.
So that's
there
maybe
within
in
your
the
brain.
late stages.
Yeah.
So then there would interfere with
Function of your brain and
normal function.
yeah, and you basically develop what's like dementia, where you kind of don't know where you are, you don't know what you're doing, you don't know what you did yesterday, you can't process.
A lot of like any kind of disease too, when you're at the end of whatever, cancer even.
Yep, cancer can move into the brain just like this.
Yeah. So a lot of
Yep, just like this.
So the BBB didn't catch the can.
In the case of our
Yeah.
discussion, yeah, no, it didn't. In this case with HIV, since it's a virus, and it's infecting your white blood cells, part of the blood, it didn't catch the HIV virus. We have HIV in its active form, so it's now moving throughout the body and it's infecting a larger number of CD4 cells. And in this particular case, it can be one of the ways that the HIV can infect the brain tissue. Because there's no BBB there, there's no protection there.
And everyone has them? Like everyone?
Everyone has a
The
B.
parts that won't have them.
Yes, that don't have they have circumventricular organs, yes. Hypothalamus, pituitary, pineal, all of those three really don't have a BBB. That's in everybody. That's true for everybody. Hypothalamus, again, this is the area that's above the, okay, now we can see the pituitary gland, which is here. And above that, here's the hypothalamus. You can see the thalamus, part of the mass of the thalamus, which is right here at the top of this. So here's the thalamus. We're going below, below the thalamus to the hypothalamus. Similar to the thalamus, when we're looking at that, there are many different areas, like I said, they're colored, they each have a job, they each have a function. It's too much information. You don't need to know all of that information. For hypothalamus, you should know that it is going to be the main regulator of the endocrine system. It's going to be the area that will control the action of the pituitary gland. It's kind of the boss of the pituitary gland. It's going to be responsible for inhibiting hormones and releasing hormones. And just overall functioning of the and overall functioning of homeostasis. I have to laugh at myself.
Function or functioning?
Functioning. I think I was that. Rewind. You do not have to know every single one of these colored areas. We will come back to the hypothalamus and the endocrine system in ANP2. And so you'll see how it controls the pituitary gland with the inhibiting hormones and the releasing hormones. So that will be fun. We'll come back to these. These are just some of the functions of the hypothalamus. As I said, it's going to regulate the pituitary gland. The pituitary gland has an anterior and a posterior portion. It's going to control and integrate the activities of the autonomic nervous system. And it's also going to play a big role in controlling your body temperature. There's so many different things. I've just highlighted a few. If you didn't watch it, you should watch it. It's a really good movie. There's some really good references in the movie. I like it. And he understood it too. And I was like, wow. I have such a smart son. Okay, so looking at the, am I going over? I probably am. I feel like I am. Yeah, I am. Okay. I only want to talk about one more thing, and then I'll give you a break, I promise. So on the cerebrum, which we are finally at, the cerebrum, because I want you to focus on this in the last portion today. On the cerebrum, there is a deep groove towards the center of the cerebrum. That's called the central sulcus. The cerebrum, in the middle of the cerebrum, is a deep groove called the central sulcus. You can see it nicely because this brain is colored. Okay. The central sulcus is in between the red and the blue gyrus here. Between the red and the blue. Gyrus. With a G. This area here is colored in red. Is called the pre-central gyrus because it's before the central sulcus. This area in blue is called the post-central gyrus. It's after the central sulcus. I'll say it again, don't worry.
Pre-central.
So here's the central sulcus in between the red and the blue. Pre-central gyrus is here in red, before the central sulcus. Post-central gyrus is here in blue, after the central sulcus. Why is this important?
Gyrus is the motor, isn't
A gyrus is a protrusion in the brain. It's a hill in the brain. So the pre-central gyrus, which is here in red, is going to be the primary motor area. When you're looking at the upper motor neurons coming from the brain, they're going to come from this area here, from this gyrus. The blue area is the sensory area. When we're looking at the first order neuron, the second order neuron, the third order neuron, and the sensory pathway, those sensory pathways are going to come up and synapse here within the post-central gyrus.
Post-central is primary sensory.
Yes. So the other colors that you're seeing here represent the lobes of the cerebrum. You have, I think, to fill out some of this in your packet. So the frontal lobe, the parietal lobe, the occipital lobe, the temporal lobe. Do you all remember that? That's what these colors are for. If I was going to ask a lobe, I would use one of the colored brains. Okay. Although, even if I used this one, you should still know that that's frontal and that's occipital. But if I'm going to ask a lobe or central sulcus or pre-central gyrus and post-central gyrus, I would use one of the colored brains like this. Questions on this? I don't want to talk too much. I want to give you a chance. Yeah. Okay. Feel free to come and take the brain models that I was using. And if you need a break, certainly take one. I will pull out some brains, some more brains. Sheep brains, I think we have.